Hello, grade 10 students. In this video, we're going to summarize the nervous system, part 1. In this slide, we're going to talk about the function of the nervous system. The nervous system has different functions. The first one is to receive the information. For example, I am in a room. I have some items in the room, so I can see them using my eyes, so I receive information. For example, I can smell using my nose, so I'm receiving information. I can hear using my ears, so I'm receiving information. This is the first function. Second, to process this information, meaning that to analyze this information to take the proper decision. For example, I see, for example, a hot cup. This hot cup, I should not approach my hand. So this is the processing the information. I process the information, the brain send an order to do it. You don't have to remove your hand or approach your hand toward this hot cup. Also, it has to respond. For example, I saw a water bottle. I wanted to drink. So I receive the information using my eyes. I see the water bottle. Second step, I process the information. I'm thirsty. I have to drink water. So here I'm processing the information. Third, I respond by approaching my hand to get the water. This is the brain and the spinal cord, which compose what's known by the central nervous system and the nervous system. As I said, is composed of the central nervous system and the peripheral nervous system. Central nervous system is abbreviated as CNS, central nervous system, whereas the peripheral nervous system is abbreviated as PNS, peripheral nervous system. Central nervous system is composed of brain and spinal cord, whereas the peripheral is composed of nerves, cranial nerves that ramify from the cranium, the spinal nerves that ramify from the spinal cord. Also, we have the brain is composed of cerebrum, cerebellum, and the medulla oblongata, whereas the cranial nerves they ramify, as I said, from the cranium, from the skull, from the brain, and the spinal nerves they ramify from the spinal cord. Cerebrum, cerebellum, and the medulla oblongata they compose the brain. They analyze the received information, they integrate them, and they take the proper decision or the proper order. Whereas the nerves they have a function of carrying the messages between the central nervous system and the effector organ. For example, they carry the message between the brain or spinal cord and the receptor effector organ, meaning that, for example, my hand is the receptor and it's also called effector organ because it has an effect and it receives the messages. Okay, so here this figure is a very important one. This diagram, nervous system, as we said, is composed of two parts: the central nervous system, CNS, and the peripheral nervous system, which is the PNS. Central nervous system is composed of brain and the spinal cord, whereas the peripheral nervous system is composed of nerves. Brain is composed of three different parts: cerebrum, cerebellum, and the medulla oblongata, whereas the nerves they are composed of cranial nerves and the spinal nerves. Cranial nerves they ramify from the brain, and the spinal nerves, as their name indicate, they ramify from the spinal cord. So cerebrum, cerebellum, and medulla oblongata they compose the brain. They analyze and integrate the messages, and they take the proper decision. Whereas the messages are carried by the nerves between the central nervous system and the receptor or the effector organ. So here you have to pay attention that the cranial nerve ramify from the brain. So cranial, brain, spinal from the spinal cord. The nervous system is a network that mainly includes stimulus, includes the receptor organ, includes the central nervous system, includes the effector organ, and finally the action or the response or the reaction. Here, the receptor organ, it has a function of recognizing the stimulus and stimulating a message. For example, I saw a cup of tea. So the receptor organ here, it's the eye. So the eye saw the stimulus, which is the cup of water, so it recognizes and stimulates a message. This message is carried to the central nervous system, but it has to be carried by a special taxi. This taxi is called the sensory neuron. A neuron conducts a message from the sensory organ toward the central nervous system through a pathway called sensory pathway. While message from the central nervous system is going to be carried to the effector organ, where the function of the central nervous system is to receive information, to analyze it, to integrate it, and take the proper decision. While the message between the central nervous system and the motor or the effector organ, it has to be carried also by a special taxi. This taxi is called the motor neuron, or it's called efferent. It conducts a message from the central nervous system to induce a response, and then the message is moved from the effector organ, which receives the decision to perform or an action or to stop this action, and finally we will the action. So you have to know that between the receptor organ and the central nervous system we have a special taxi. It's called the sensory neuron afferent. While between the central nervous system and the effector organ we have a special taxi which is called motor neuron if. Here in this figure, the man observes the stimulus, which is the cup of water, using the eyes. Then a sensory message is sent from the eyes toward the brain, which is the central nervous system, for integration. And then a message is sent from the brain to the muscle to take the response. So the message from the eyes toward the brain is sent through the sensory input through the afferent neuron, whereas the message from the central nervous system toward the hand is transported by the motor input, which represent the motor neuron, which is the efferent neuron. Output. So cup of water represent the stimulus here. The stimulus here is the cup of water. So we see the cup of water through the use of my eyes. Then this is the sensory organ. Sensory organ. Then it moves to the central nervous system by the help of the sensory neuron. As we said, it's called afferent neuron. Then it moves to the central nervous system where it's integrated and sent from the central toward the muscle of the hand by the help of the motor neuron is called efferent. Some of you is going to ask me, how can we differentiate between afferent and efferent? They are very close to each other. So the first one is the sensory. Sensory afferent. Why? Because letter A starts before the letter A. So this is a way you can differentiate between afferent and efferent. So always we sense before we have the motor action. So sensory first, the letter A is the first letter, so we can say that sensory is afferent and not efferent. Then the muscle of the hand, they are called the receptor or the effector organ, and then the hand approach, and we have the response or we have the reaction. Here is the response approach of the hand. Okay, so stimulus number one, this is the pathway. Number two, we have the receptor organ. Three, central neuron. Number four, central nervous system, and inside the central nervous system we have special neuron. They are called interneuron. We're going to talk about them later. Number five, the motor neuron. Number six, the effector organ, and number seven, which is the last step, we have the response. This is the pathway of the nervous message.
Let's move now to the anatomy of a neuron. This is a neuron. The neuron is composed of different parts. First, the dendrites. These are the dendrites. The cell body, or it's called the soma. This is the cell body, or it's called the soma, that contains the nucleus. This is the nucleus, the one in blue here. And the axon, this is the axon, the one in blue. It may or may not be myelinated. We will discover the role of the myelin sheath next. Also, we have the terminal arborization, or the lobes, or they are called the terminal buds.
So here, these are the dendrites, hair-like structures. Here, the nucleus. This is the cell body, is called, as I said, it's called the soma. Here, the myelin. The blue one is the axon. So the axon is this one, the blue one here. This region, this is the axon, and here the synaptic terminal. So neuron basic structure unit of the nervous system. Dendrite, they carry impulses toward the cell body of the neuron, and the axon carry impulses away from the cell. Myelin sheath has a different role. Synaptic terminal contains many neurotransmitters. They contain epinephrine, norepinephrine, acetylcholine, and GABA. All of them, we're going to talk about them in the last stage of this chapter. So a group of nerve fibers, they form a neuron. A group of neurons, they form a nerve. Group of nerves, they form what's known by the nervous system.
So in this figure, we have the difference between the afferent neuron and the efferent one. The afferent neuron, it carries the message from the sensory neuron toward the brain, which is the central nervous system, while the efferent, it carries it back from the brain toward the effector organ to take the proper action.
So the structure of the neuron, here we have the myelin sheath. Myelin sheath, it acts as an insulator, and also it has a function in acceleration of the nervous message. Why? The nervous message in normal cases, it moves in this way along the axon, while in the case of myelin sheath, the nervous message has to move over the myelin sheath, so it has to jump in between these spaces between each myelin. These spaces, they are referred to as node of Ranvier. So that's why the message in the myelinated nerve fiber, or the myelinated axon, the message is going to be faster because it's going to jump. This jumping is called saltatory movement. So myelin is a lipoprotein that acts as protective layer of the axon and also as an insulator. It's not continuous. The gaps, as I said, they are referred to as nodes of energy. Actions, they are classified as either voluntary or involuntary. From now on, we have to replace the word involuntary by the word reflex. So voluntary actions, these actions require thinking. So it has to intervention of the brain and the spinal cord. For example, I have to hold my phone. Holding my phone is an action that require thinking, while the reflexes, the action that do not require thinking and do not require the intervention of the brain. For example, I hold a hot cup of tea. I hold it. I thought that it's normal. It's very hot. So immediately I'm going to throw it. This action, or this reflex, is called the reflex action. It doesn't require the intervention of the brain, and it doesn't require thinking. How to measure the speed of the propagation of the nervous message? Measuring the speed of propagation of the nervous message, we have this experiment, stimulating electrode, and in this stimulating electrode, we have both of them, they are placed inside the axon. delta D, the distance between these two stimulating electrodes, and we have the reaction. How can we measure the speed? The speed is delta D over delta T. Delta D is the distance. This one, it's the distance over delta T, which is the time. Okay? Factors that affect the speed of the propagation of the nervous message inside the nerve fiber. Diameter of the nerve fiber. As the diameter increases, the speed of the propagation of the nervous message will increase. So D1 is less than D2. So D2 or V2 is greater than V1 due to the larger diameter. So diameter here is larger, so speed is higher. Okay, so as a conclusion, as diameter of the nerve fiber increases, the speed of propagation of the nervous message also increases. Myelination of the nerve fiber. Myelination of the nerve fiber, the presence of myelin. As we have myelin, sure, the speed is going to be higher in the case of myelin. So V2 for the nerve fiber 2 is greater than V1 for fiber 1. So as a conclusion, the presence of myelin has higher effect on the increasing the speed of propagation of the nervous message than the effect of the diameter. Fiber move straight. Why? The case of myelin is going to move in a jumping manner, which is known by a saltatory manner. So myelin is an insulator. It doesn't contact a nervous message. So it moves like this in a saltatory movement by jumping. So the nervous message propagates in a saltatory manner. Is the neuron always active? Are we always listening? Are we always walking? Are we always talking? Are we always in pain? Sure not. So here, a neuron and a nerve fiber is said to be at rest in the absence of any stimulation or in the absence of effective stimulation. And pay attention, effective stimulation. We're going to explain this in the coming parts of our chapter about the nervous system.
So now let's move to the series of these experiments where their aim is to study the electrical state of a neuron at rest. So experiment number one, we have the nerve fiber, the axon, which is placed inside a physiological solution where the terminals of the voltmeter and the oscilloscope, they are connected to the external medium of the axon. So the needle of the voltmeter didn't deviate from zero. What does this mean? This means that there is no potential difference, there is no delta V potential difference across the surface of the axon, or you can say that there is no potential at the surface at all. Experiment number two, the needles of the voltmeter, for example, the simulating electrodes here, they are placed inside the axon where the needle of the voltmeter didn't deviate also from zero, and the oscilloscope shows that delta V is equal to zero. What does this mean? This means that there is no potential difference across the surface of the axon or the internal medium. Moving to experiment 3. One on the internal side and the other in the external. This one is the internal sides and this one on the external side. The voltmeter, the needle deviates to minus 70, and the oscilloscope shows the delta V is equal to minus 70 millivolt. So what can you use here? We can deduce that there exists a potential difference between the internal and the external side of the nerve fiber, where delta V does not equal to zero. As such, at rest the nerve is polarized, the nerve fiber is polarized, is negative. Since delta V is less than zero, then the interior of the nerve fiber is negatively charged with respect to the exterior. Note that the existing potential is the potential difference that exists between the intracellular and the extracellular media of a nerve fiber that is at rest. Resting potential, there is an electric charge across the membrane. This is called the membrane potential. Resting potential when the cell is not firing means that when the cell is at rest, it's a minus 70 millivolt between the internal and the external side. So have to note that resting potential may range between minus 5 to minus 90. This is depending on the species and the nature of the nerve. Ions, they electrically charged molecules. For example, sodium positively charged Na+, potassium K+, chloride Cl-, negatively charged. The resting potential, it exists because ions are concentrated on either sides of the membrane, where Na+ and Cl- they are outside the cell, and K+ and other organic anions inside the cell. So here, concentration of the ions, for example, concentration of Na plus outside the cell is sometimes 10 times greater than this concentration of Na plus, 150 millimolar. Inside the cell, Na plus 15 millimolar. It's about times 10. So how is resting potential maintained? Due to differences in the membrane permeability or selective permeability, some ions to sodium and potassium. Due to differences in ion concentration between the intracellular and extracellular, and due to the action of the sodium potassium and active pump Na plus K plus pump. To keep in mind what the meaning of an excitable neuron. A neuron that's able to respond to an effective stimulus. Action potential, it's the term given to the electric visualization of a response on neuron. For example, whenever you see this means we have action potential. Also, to keep in mind what the meaning of the minimum time of excitation. The minimum time of excitation is the minimum time during which an effective stimulus should be applied in order to excite a neuron. So action potential in a nerve fiber at rest, minus 70 millivolt, it's called potential. When I have an application of a stimulus of intensity I1, it's less than the threshold of excitation. So your I1, there is no change in the potential difference. So this is said to be ineffective stimulus. While you're applying intensity I2, which is also less than the threshold, but it's greater than I1, we have a small increase in the potential difference, but it didn't reach the threshold of depolarization. So here it's called artifact, or it's called hypopolarization. That didn't reach the threshold, so everything will go back, and this stimulus is said to be ineffective. While after application stimulus intensity I3, which is greater than or equal the threshold. So I3 is greater than I2 for sure. So here we have action potential is up. So enough change in the potential difference that reach the threshold of depolarization, and action potential or response of amplitude 120 millivolt was recorded. As such, the stimulus of intensity I3 is said to be effective and said to be the threshold of excitation. While I4 greater than the threshold and is greater than I3, so we will have the same action potential. This is called the effective stimulation. So here are the phases of the action potential in nerve fiber. where phases 1, 2, 3, 4, 5, 6, and 7. So number 1, it's called the resting. Number 2 is called hypopolarization or the artifact. Number 3, going to the positive, depolarization. Number 4, inversion of polarity. Number 5, repolarization. And number 6, hyperpolarization. And number 7 is going back to the resting state. So resting phase, resting potential, hyperpolarization or the artifact, depolarization, inversion of polarity, repolarization, hyperpolarization, and returning back to the resting potential. Here we have this figure where resting potential we have polarization, then we move to the depolarization where the Na plus channel is open. Phase number 3, the repolarization where the K plus channel is open, and then we go back to the phase which is the resting phase. So you have to know that if there is a weak stimulus, there is no action potential. To reach the threshold, we have an action potential. Strong stimulus, it will have more than one action potential, but as you see, all of them they have the same amplitude. So the message here obeys the law of all or none. Either we have an action potential or not, but what differs is the frequency, how many times it repeats itself. So here you have to know that the message propagates in a unidirectional manner from the dendrite or the cell body to the axon and then to the next cell, dendrite, cell body, axon.